Okay, ji, today we should discuss about the role of electric forces on ion movements. Uh, Yesterday we had discussion about the various other factors which were controlling the diffusion. For example, we have discussed about the direction and magnitude of the solute. Similarly, solute concentration differences, as well as we have also discussed about the solubility in the membrane. And as I have told you that the composition of the membrane and the composition of the uh, solute molecules uh, uh, that are very much important. For example, when we talk about the structure of the plasma membrane, it is basically uh, lipid bilayer membrane. It is having lipid in the form of the bilayer, and that's why we have said that non-polar molecules or the lipid soluble particle, they can move across the membrane easily as compared to the uh, polar molecule or the water soluble molecule. And similarly, we have also discussed about the uh, permeability of the polar molecule, and I have told you that these polar molecule, and specifically those molecules which are having ions or which are dissociated into the ions, those ions, they may cross the membrane with the help of certain types of the uh, proteins which are called as ion channels, and they are having a specific size and specific diameter, as well as specific electric composition to allow the movement of the certain ions through them. Can you hear me clearly?
Yes, sir.
Yes,
Okay.
sir.
So when we describe the diffusion of the ions, as I have told you that they are the particles, in addition to the size and in addition to the composition of the ions, as well as the composition of the ion channel. Composition in the sense the electric charges which are present on the ion, whether the ion is positively charged or it is negatively charged. That's what we can say the composition of the ion. Another additional factor that should also be in consideration, that is electric force which is acting on the ion. This is another important factor, that which type of the force is acting on the ion, that will also decide the movement of the ion across a membrane. So as all of you know that there exists a separation of the electric charges across a membrane. The plasma membrane is always having a difference of the charges, separation of the charges on the two sides of the membrane. Mean two sides of the membrane have the same charges. The two sides of the membrane will always have different charges. Mean sometimes the outer side may have the positive charges and the inner side may have the negative charges. Or some other time the outer side is positively charged and inner side is negatively charged. So there will always exist a difference of the charges on the two side of the membrane, and their difference of the charges is called as membrane potential. That is called as membrane potential. Okay, so here we can see this slide. This is a plasma membrane, and this is as extracellular fluid. On the extracellular side, you can see that there is bulk of the positive charge. Positively charged ions are the positive charges. They are present on the outer side of the membrane, and negatively charged ions are negative charges. They are present on the inner side of the membrane. So difference of the charges on the two side of the membrane, that is what we can call as the membrane potential. And here in this case, you can observe that negatively charged particles, they are moving towards the outer side, and positively charged ions, they are moving towards the inner side. How their movement is accomplished? As we have discussed in previous lecture, that these ions basically they move through the ion channels. which are present on the surface. So for example, this is sodium ion. This sodium ion will move through this ion channel. This is potassium, this is chloride ion, or any negatively charged ion, it will move through this ion channel. So separation of the electric charges across a membrane, that will be called as membrane potential, and it actually provides the electric force. What we mean by electric force are the electric current. And electric current is basically the charge difference on the two what we can say there are two terminals, or the two, one is anode, another is cathode. So there would be a charge difference. One end may be having the positively charged ions, and the other end will have the negatively charged ion. So these two opposite charges, they will attract each other, as a result of which current will pass through that particular surface. So this is what we can call as membrane potential, and this provides the electric force, and this electric force will drive the positive ions inside the cell and negative ions outside the cell. Due to this charge separation, an electric force will be generated, electric current will be generated, and this electric force will move the positively charged ions to the inside of the membrane, and the negatively charged ion towards the outside of the membrane. So electric charges of the same sign, both positive or both negative, if we are having same charges on the two sides of the membrane, for example, and similarly in front of this negatively charged ion, if there is another negatively charged ion, these two may repel each other. So there should always be a difference of the charges, separation of the charges, and through this membrane the electric current can only pass when there are different charges on the two sides of the membrane. So electric charges of the same sign, whether they are positive or negative, they will repel each other, while opposite charges, they will always attract with each other. So if we talk about the inside of the cell, if inside of the cell is having a negative charge, and with respect to the outer side, as in most of the cases, the outer side of the cell uh, is is positively charged, whereas the inner side of the cell is negatively charged. This is most of the cases. So an electric force will be generated, as I have told you, that this charge separation will provide an electric force, and this electric force will attract the positively charged ions or positive ions into the cell, and repelling the negative ions. So these if positively charged ions, they are moving inside the cell, so the negatively charged ions, which are already present inside the membrane, they will repel each other, whereas they will attract the positive ion which are on the outer side of the membrane, and hence a current will be generated. So if there is no difference in the ion concentration across the membrane, still there will be a movement of the ion. What we mean by difference of the charges, mean, for example, if we have 100 positive ion on the outer side of the membrane, and 100 negative ion on the inner side of the membrane, so there is no concentration difference. Both side of the membrane is having same number of the charges. So this is what we can call as uh, concentration difference. But as in case of diffusion, we all know that for the diffusion process to occur, there should always be a concentration gradient. Mean there should always be a concentration difference on the two sides of the membrane. Mean one side of the membrane should always have higher concentration of the solute, and other side should always have the lower concentration of the solute. So in this case, the diffusion process would be possible. But if we take this scenario, in this case, the concentration of the solute, or the concentration of the ions on the two sides of the membrane, that will not matter. Suppose we have equal number of the positive ion and negative ion on the two sides of the membrane, still the movement there would be some sort of the movement across a membrane. So still there would be a net movement of the positive ion into the cells and negative ions outside of the cells, because of the potential difference, or the membrane potential. Now there is no concentration difference, but there is still the potential difference. There is difference of the charges on the two sides of the membrane. So in spite of having same concentration of the ions on the two sides of the membrane, still due to the difference of the charges on the two sides of the membrane, still there will be net movement of the positive ion from outside to the inside and negative ion from inside to the outside. So direction and magnitude of the flux, ion flux across the membrane, it will depend upon the concentration difference and the electrical difference, which is also called as membrane potential. So although the movement will take place, but how we will determine the net flux and magnitude mean direction of the movement of the ions and the intensity of the ions will be dependent upon the concentration difference as well as the electrical difference, which is also called as membrane potential. So here two driving forces are there. One is concentration gradient and other is membrane potential or the potential difference. So these two forces collectively they will be called as electrochemical gradient. These two forces collectively they will be called as electrochemical gradient. It can also be called as electrochemical differences across the membrane. So it is very much important that these two forces they should oppose each other, mean they should be for example, we have said that positive charges are on the outer side and negative charges are on the inner side. So it means this membrane potential they are opposing each other, or they are on the two different sides. Similarly, if one area is having higher concentration and other area is having lower concentration, so at the same time here you can say that still there is potential different concentration difference will also exist there when we take these ion individually. For example, when we talk about these positive ion, they are having higher concentration on the outer side of the membrane and lower concentration on the inner side of the mean at the very initial stage there is no positive ion on the inner side, so there is huge difference of the concentration on the two side of the membrane. So definitely the concentration gradient will favor the movement of the positive ion from higher concentration to the lower concentration. Similarly, when we talk about the negative ions at this specific moment, all the negative ions or negatively charged ions, they are present on the inner side of the membrane, and there is no single negative ion on the outer side of the membrane. So it means there is a huge concentration difference. So at this particular time, the concentration difference will also favor the movement of negative ion from higher concentration to the lower concentration. So one is charge difference, which is called membrane potential, and other is called concentration difference. These two are collectively called an electrochemical gradient, and these two forces they will in a combined way they will affect the movement of the ion. They will decide the movement of the ion, whether the ion they will move from outside side to the inner side, or they will move from inner side to the outer side of the membrane. So they should always uh, oppose each other. So membrane potential, for example, let's we take an example of potassium ion. In one direction, the membrane potential is taking the potassium ion across the membrane, while the concentration difference for potassium is driving the ions in the opposite direction. So it means uh, electrical potential or the membrane potential is driving the potassium ion in one direction, and the concentration gradient or the concentration difference that is driving in the opposite direction. So it means these two forces, one is called membrane potential or the potential difference, and other is called concentration difference or the concentration gradient. These two forces they will act in opposite way. They will act always in opposite direction. So net movement of the potassium in this case, that will be determined by the magnitude of these two opposing forces by the electrochemical gradient across the membrane. So net movement in this case, as in this case, we can see here that the net movement will be determined by the magnitude, as we have discussed that the net flux is actually the difference of the two one-way fluxes. So in the similar way, the net movement of the ions or negatively or positively charged ions are the molecules that will always be determined by the difference of the potential membrane potential as well as the difference of the concentration gradient. So ion channels they can be present we have already discussed that they can be present in a donut form. Mean they can have an open configuration, or sometimes they can have a closed configuration. So changes in the membrane permeability to ions that can also change their opening or the closing state. Mean when there is change in the membrane permeability, the ion the conformational changes in the ion will also take place, and they may assume their open configuration, or they may assume their closed configuration. So this process of opening and closing of the ion is called as channel gating. Just like a gate in our house, in our home, we open and close that gate. In the similar way, the ion channels they may remain sometime in the open configuration, and some other time they may remain on the closed configuration, depending upon the permeability condition of the membrane to a particular type of the ion. So this process is actually called as channel gating. So channel sometime may open and sometime they close. Many times, even during a single setting, these channel can have open configuration as well as they can have closed configuration many times. So the channel protein actually fluctuates between two conformation, in between the open conformation as well as in between the closed configuration. So if we talk about extended period of the time, any at any given electrochemical gradient, total number of the ion that are passing through a channel, they are actually dependent upon the frequency of the channel open. Their movement will be dependent upon how frequently the channels they are open and how long it stays open. So two important things are there. One is channel opening, and for how long period they actually remain in open state. These are the two important factors for the movement of the ions across a membrane. So in 1980, a technique was developed to monitor the single ion channel. Let's take this figure as an example, and in this figure we are taking a single ion channel to monitor the movement of the ions across the membrane. So these are actually the channel protein which are behaving as the ion channels, and through these ion channels the molecules or the ions which are having positive charge or the negative charge, they are actually moving across this ion channel. So this technique is called as patch clumping technique, and in which we actually place the tip of a glass pipette on a small region of the cell surface, and we apply the section to that glass pipette, so that the membrane patches become sealed to the edge of the pipette. So when you put the glass pipette on the surface of any cell which is having the elastic membrane, and you apply the section to that particular area with the help of the glass pipette, so that area of the membrane will become we will stick to the end of the glass pipette, and so that it will remain attached when the pipette is withdrawn from that particular. This is called patch clamping. So ions, since they carry an electric charge, the flow of the ion through an ion channel in the membrane patches, it will produce an electric current. And now this electric current that can be monitored. Here in this example, we can observe the two conformation of the channel protein. Either they are in closed conformation, or they are in the open conformation, or they are in the closed conformation. These are the two states of the ion channel. So this conformational changes in the protein, which is actually forming an ion channel, it can either be in open form. This is the open form, and this is the closed form. This is the extracellular side, and this is the intracellular side. As you can see here, that the two sides of these ion channel, they are separated by the phospholipids, or the lipid bilayer of the plasma membrane. So current flow that was uh, basically that was intermittent, mean that was not continuous flow. It was corresponding to the opening and closing of the ion channel, and the current magnitude was measured as the result of permeability to that particular ion. So when possible inhibitors were added there, the stimulants, they were added in the solution, in the pipette, or in the fluid, which is actually in contact with the intercellular surface of the membrane. We can effect, or we can analyze the effect of these agents in modifying that, how the frequency of and duration of the channel opening that was altered. So initially, the section was applied on the surface of the channel protein with the help of glass pipette, and later on, inside the glass pipette, certain stimulants or certain inhibitors were added to observe the frequency and duration of the channel opening, whether after addition of the inhibitors or the stimulants, now the frequency of the channel opening and the duration of the channel opening, whether that is altered or not. Mean frequency, how many times in a single second the channel they open, and duration mean how long it remained open during a single second. This is what we can call as the frequency as well as the duration of the channel opening. So with the patch clamping technique, this duration as well as the frequency was monitored, and it was actually to observe whether there was any alteration in the frequency, any modification in the frequency and duration of the channel opening by addition of certain inhibitors or the stimulants. So patch clumping basically help in following the behavior of a single channel over time. That how a single channel actually behaves when a section was applied, or when current was generated on that particular area, and how it behaved, how many times it opened, and how long reaction. So three factors. There are three factors that are very much important for channel protein conformation, which can affect the channel protein conformation, and they can produce the changes in the frequency or the duration of the channel opening. So one important thing is binding of specific molecule to the channel protein may directly or indirectly produce an allosteric or covalent change in the shape of the channel protein. For example, if here any particular molecule, it may bind, let's suppose in this particular area, or in this area here, if any particular ion or molecule which is other than the ion channel, ion which is specific to bind with this ion channel. So if any other molecule, it may bind on the surface of this channel protein, there will be conformational changes, or the allosteric changes, or the covalent modification on the protein, and hence its open or closed conformation or configuration will be changed. So one important thing is binding of any specific protein or any specific other molecule which is other than the ion which is passing across the membrane. It will result in allosteric changes, the conformational changes in the structure of the channel protein. These channels are actually called a ligand sensitive channel. Ligand is any type of the chemical, any type of the messenger. When this messenger or chemical binds to the surface of this channel protein, it will result in allosteric or the covalent modification on those channel protein. So those channels are actually called as ligand sensitive channels, because they are very much sensitive for binding of any type of the ligand or any type of the specific chemicals. And the ligand that influence them, they are most of the time they are called chemical messenger. Messenger, as all of you know, it can either be hormone, autocrine, or paracrine. Binding of a lesion. The second important thing is the changes in the membrane potential. They can cause the movement of the charged regions on a channel protein, and that can also alter its shape. Those type of the channels are called voltage gated channel. So first type of the channels are actually called as region sensitive channel. They change their shape, they change their open or closed configuration as a result of binding of any specific chemical that is called ligand, that is called messenger. And then the second type of the channels are those channels which are actually sensitive to potential difference. When there is change in the voltage or change in the electric current on the two side of the membrane, these channels may also change their shape. They may change their shape from the open configuration to the closed configuration, or sometimes they may change their shape from the closed configuration to the open configuration. So second type of the channels which due to change in the membrane potential difference or the electric current, those channels are called voltage gating channels. So second type of the channels are voltage sensitive channels or voltage gating channel. And the third type of the channels are basically those channels which can be affected by physical stimulation. For example, we stretch those channel or we stretch the area of that membrane by any type of the physical effect. Those channels are called mechanosensitive channels. They may be stimulated, they may be affected by applying the pressure, by applying any type of the physical stimulation. So sometimes the single channel may be affected by more than one of these factors. Sometimes the single channel can be affected with more than these factors which we have discussed. For example, we have discussed about the ligand sensitive channel, we have discussed about the voltage gated channel or the voltage sensitive channel or mechanosensitive channel. Sometimes these three factors simultaneously they can affect a single ion channel as well. So particular type of the ion which is actually moving across the membrane or which is moving through different types of the channel. For example, if we take a single membrane, this single membrane can either have it can have ligand sensitive potassium channel which is called K channel. It can also have a voltage sensitive potassium channel, and it can also have mechanosensitive potassium channel. So it means the ion is single. The ion is only the potassium ion, but on the surface of the membrane, three type of the channel can be present. Those channel can either be ligand sensitive, those channel can be voltage sensitive, as well as those channel can also be mechanosensitive channel. So for a single type of the ion, sometimes the membrane can have three different types of the ion channel, and this ion can pass through any of these three channels. This ion is having the luxury of passing through any of these three channel. Either it can pass through the ligand sensitive potassium channel, it can also pass through the voltage sensitive potassium channel, or sometimes it can also pass through the mechanosensitive potassium channel. So this is one important point. The second important point is sometimes the membrane can have several type of the voltage sensitive potassium channel. Mean in first case the membrane can have single voltage sensitive potassium channel. In another case the single membrane can have several type of the voltage sensitive potassium channel, and if this voltage sensitive different number of the voltage channel, they may respond to different range of the membrane potential, and several type of the ligand sensitive potassium channel or several type of the mechanosensitive potassium channel, they can also be present, and those are not specific for a specific ion. They can have permeability to certain other ions as well. And in the second case, if we have several type of the ligand sensitive potassium channel, those ligand sensitive potassium channel can respond to variety of a ligand. For example, they can respond to a large number of the different types of the hormones or the messenger. So that's all for today. Okay, any question? If you have any question, you may ask, please. जी, एनी क्वेश्चन? समझ आ गई सब लोगों को? कैन यू
Yes, sir.
हियर मी? Any question, please? If you have any question, you may ask.
सर हम लोग आपकी
जी,
रिकॉर्डिंग कर सकते हैं, आपने वहां से म्यूट किया होता है, यहाँ पे तो म्यूट का ऑप्शन आ रहा है आपकी तरफ से।
किसका?
आपकी स्क्रीन पे मीन्स म्यूट का ऑप्शन आ रहा है, हम आपकी स्क्रीन रिकॉर्ड नहीं कर सकते, लेक्चर रिकॉर्ड नहीं कर सकते।
वो मैं कर रहा हूँ ना, अगर आप लोगों को चाहिए होगा तो आई कैन प्रोवाइड यू। मैंने
सर वो
म्यूट नहीं किया हुआ, ये कोई ऑटोमेटिक शायद ऐसा है, मैंने
जी,
म्यूट नहीं किया हुआ।
अच्छा सही है, ठीक है।
ठीक है, पहले
ओके
मुझे
सर।
यहाँ से बंद करने दें, फिर आप ट्राई करें। अब आप इसको ट्राई करें।